Hi everyone, thanks for coming. My name is Moreira. I'm going to talk about the research we've been doing at the American Museum of Natural History on the genomics of North American woodpeckers. So a long-standing question in evolutionary biology is the relative importance of determinism and stochasticity in adaptive evolution. And a way of addressing this question is by looking at cases of species or populations that have repeatedly adapted to the same environments. And one of the classic examples is the sticklebacks that have adapted to the freshwater environment multiple times to the recruitment of the same alleles. But we also have examples of species that have adapted to the same phenotypes through different genetic mechanisms, which is the case of pocket mice who evolved melanism through mutations in different genes. So I'm studying parallelism in the hairy and downy woodpecker, which is a classic avian system in evolutionary biology. If you're not familiar with the hairy and downy woodpecker, they are widespread species in North America. You can find them basically anywhere you find trees in North America. They're a classic example of convergence in plumage, because even though they look very similar, they are not related, they're actually quite distant from each other. They also show parallel geographic variation in plumage and body size. So usually you have individuals that are darker in the west and lighter in the east, and larger in higher latitudes and elevation as well. They also show similar Pleistocene dynamics. So during the last glacial maximum, the populations of both species were restricted to southern refugia, and after the end of the Pleistocene, when the glaciers retreated, these populations expanded their populations north, recolonizing and readapting to these northern environments. So currently, they occur along a huge environmental gradient from the cold border forest in Alaska to the subtropical forest in the southeast. And accordingly, their phenotypes seem to have evolved in response to these climates. So here's a recent study by Cooper et al., where they found a significant relationship between body size and latitude in both species, as you can see in this graph. So given this amazing diversity of phenotypes and ecology, we don't know how single species could adapt to such a big range of conditions. So with the study, we were trying to understand first how genetic diversity is distributed in these two species and how they responded to past climate change. And we are also, interested in understanding if we find evidence for signatures of selection in both species, and if those signatures of selections occur in parallel. So to do that, we obtained genetic samples from the field as well as from museum loans. We sampled 10 individuals per population in each of 7 geographic regions in North America, and these geographic regions represent major climatic domains in North America. And these goals I'm going to use to refer to these populations throughout my talk. We then did a whole genome resequencing to a coverage of around 5x, which generated about 34 million reads per sample. We then mapped these reads to the reference genome of the downy woodpecker using BWA, and we produced and we called SNPs using GAPK, and that produced around 17 million SNPs for the downy woodpecker and around 54 million reads for SNPs for the hairy woodpecker. Since we have low coverage data, we also did estimated genotype likelihoods using ANGSD. So the first thing we looked at was neutral population structure. So previous studies based on Sanger sequence show that in both species there is very shallow divergence among populations. And we were very excited by the resolution that the whole genome data provided us. So here's a PCA of more than 30,000 linked SNPs in the downy woodpecker. And the first thing you can see here is that PC1 basically separates populations into 3 groups. So you have Alaska here, you have Eastern plus Northwest in the middle, and you have Rocky Mountains here. PC2, on the other hand, separates Alaska and Rocky Mountains from everything else. And PC3 separates populations within this Eastern group. We also found that it was interesting that some of these PC axes corresponded to phenotypic variation that we see in the species. So for example, PC2 corresponds to variation in body size, and PC3 corresponds to variation in plumage. We see a very similar pattern for the hairy woodpecker, but here the northwest population is more closely related to the Rocky Mountains population than to the eastern population. So basically, PC1 in the hairy separates a western and the eastern clade. And again, you can see that here PC2 separates Alaska, and PC3 separates the northwest population. So another way to visualize this information is by looking at how genetic diversity is distributed in space. So this is coefficient of ancestry, and you can see again that in the hairy, the down woodpecker is very congruent in terms of space. So we've seen that genetic diversity is congruent in the two species, but what about demography? So here we have an SMC++ analysis. So this method is trying to estimate changes in population size over time from the present to the past. So here you have generations, and here you have effective population size. And the first thing you can see here is that all populations are expanding. And that expansion seems to match around the last glacial maximum. But the timing of the expansion also varies among populations. And you see the populations that are further south tend to expand earlier. We see a very similar pattern in the hairy woodpecker, but here expansion seems to occur before the last glacial maximum. And when we zoom in into each of these individual populations, you can see major differences in genetic diversity. So overall, we see that downy woodpecker shows larger genetic diversity than hairy woodpecker, and we see that eastern populations tend to show more diversity. Eastern populations, tends to show more diversity than western populations, which might indicate that these populations have been more stable over time. So okay, so we looked at genetic diversity, we looked at demography, but can we find evidence for natural selection to these different climates in North America? So the first approach to do that was to do an FST outlier analysis. So here you have, so we consider an FST outlier any window, 50 kb window, that was five standards deviation above the mean. And here we have one example comparison between Midwest and Southeast. The first thing you can see here is that the landscape of divergence between the two species is very different. So in the down woodpecker, you have way many more outlier loci than in the hairy woodpecker. You can also see that the mean divergence between the populations is very low, so it's very shallow divergence. We're just starting to look at the functions of these genes that are in this outlier peaks, but we narrowed down to a couple biological processes that might be associated to local adaptation to climate. We did that by extracting the genontology terms from the annotation using BLAST2GO. And you can see there's a variety of biological processes from brain development to sugar metabolism. So the problem with this, this is one comparison, right, for one pair of population. So we've done that for all population comparisons. And again, you see the same thing. You see the downy woodpecker show many more outliers than hairy woodpecker. So for example, if you look at Southeast, you see like many comparisons where you have a lot of outliers. And in the hairy woodpecker, you see many that have no outliers at all. So the problem with FST outlier analysis is that you're not actually looking at the agent of selection. So as a second approach, we tried to identify SNPs that were highly associated with environment. So to do that, we separated biofilm data into two sets, variables that were related with temperature and variables that were related with precipitation. Then we've done a PCA and extracted the three first principal components, which explain more than 90% of variation. Then we try to find SNPs that were highly associated with these principal components after accounting for the underlying population structure using LFMM. These are the results. So here in each circle you have the number of SNPs that are associated with either temperature or precipitation. And in the center of the Venn diagram, you have the number of SNPs that overlap between these two species. So the first thing you can see here is that there's a lot of SNPs that are associated with temperature and precipitation, which indicates that these traits are highly polygenic, as expected. We can also see that, different from the FST outlier analysis, here hairy woodpecker show many more SNPs outliers than downy woodpecker. And we also see that precipitation in general shows more association with SNPs than temperature. But the thing I wanted to focus here is the big number of outliers of candidate loci that are over the overlap between the two species, which might be indicating parallelism. So the question is this number of SNPs generated just by chance? Can they be explained just by chance? So to answer that question, you can actually calculate the probability of observing that specific number of overlap as a random sample from all your SNPs. And to do that, just calculate that under a hypergeometric distribution. And what you see here is that for both temperature and precipitation, you see that the number of sharing falls outside of the expected distribution, indicating that yes, there seems to be parallelism occurring in the system. And there might be some key genes that have unique functions for adaptation to climate in both of the species. So to summarize, we revisited this classic avian system of two species that have coexisted for millions of years, just to try to see if there is, to what extent their demography and adaptive history occurs in parallel. And we can see that despite the shallow population divergence, we can, with the whole genome data, we can find fine scale population structure. We see that all populations show strong features of expansion that matches with the last glacial maximum. Genetic diversity in these two species is very congruent in space. And we see that despite the different landscape divergence in their genomes, we can see that there's parallelism in what genes they're actually using for adaptation. So with that, I have a lot of people to thank, and I'll be happy to take any questions.
For your initial PCA looking at genetic structure, you chose a very small fraction of the SNPs that you had. Could you describe how you chose those?
Yeah, so those SNPs, the reason I have many less SNPs than my overall data set is because of missing data. So for PCA is affected a lot by missing data.
Okay.
So I try to find SNPs that are occurring across all my, and I have a lot of individuals, so I have 70 individuals. So that reduced a lot my total number of SNPs.
Lucas, did you find any candidate genes that may be associated with the color pattern variation?
So yeah, that's the next step actually. I'm gonna look for what's happening under those peaks and what are those genes that are, there are many genes associated with environment, right? So phenotype and plumage might be one of the genes. So that's the next step. I'm gonna look for specific genes that are associated with plumage.
Thank you.
Are these species sister to one another?
That's actually a really good question, because the fact that these species are so popular in the ornithology circuit is because they're actually not sister. They're thought to be sister for a long time, but they're not sister. They're in different clades, and they just evolved to be very similar. Yeah, and there's a lot of hypotheses why, which I'm not gonna go into detail yet.
Why did you choose to do whole genome sequencing rather than sequencing?
So the reason why we chose to do whole genome sequencing is because we thought that maybe a lot of the genetic basis for this very complex traits like adaptation to climate might be regulatory, and in other approaches would be hard to pick those regulatory elements.